Okay, so this is a screencast to basically go over the material that we didn't have time to go over in class today, and I just wanted you to have it available to you because the Galapagos Islands finch case study is about an adaptive radiation, and so we want you to understand what an adaptive radiation is, and we need to understand what speciation is in order to understand what adaptive radiation is. And before we start on that, we have to have some idea of what a species is. So the learning goals for this is to have you be able to explain different species definitions, so know what the different species definitions are and kind of what their utility is and when they work and are applicable and the conditions under which they're not really workable or applicable. Explain the mechanisms by which species form, so the different models of species formation, and then to explain what adaptive radiation is and how it proceeds. So we kind of went through this in class. So you have plethodonid salamander species here that looks in one way, and then you have another plethodonid salamander species that has a different form. And anybody looking at these things would say, well, these things are different, so that they might be different species. This would be invoking what's known as the morphological species concept. If two organisms are different morphologically enough, then they are different species. But that's actually not the most common species definition that you see in textbooks. Oftentimes, the textbook definition for a species is the following: a group of actually or potentially interbreeding populations which are reproductively isolated from other such groups. This is Meyer's 1942 biological species concept, and it's invoked a lot because it kind of makes intuitive sense. If two organisms are capable of breeding, they must be in the same species. If two organisms are incapable of breeding or would be capable of interbreeding, if they could come into contact with another, they are species, and if they're not capable of breeding, then they wouldn't be species. But the problem with this is that it doesn't apply to all things. So, for example, here we have two species of dinosaurs, and we can't test whether or not these organisms are different species because all we have is fossil evidence for them. And so we have to fall back on the morphological species definition if we're going to actually assign these animals to species categories. So morphological species are basically groups of individuals that are phenotypically similar to one another. But this breaks down because we have these two species, for example, the two species of gray tree frogs, and they look virtually alike. They look kind of like bark. They have a light spot under the eye, although it's a little hard to see on this one on the right. But when you listen to their mating call, it becomes apparent to you that these two species are different species. So here is the mating call of one species. And here's the mating call of the second species. And you see the trill rate is much slower with Hyla versicolor than it is in Hyla chrysosceles. And as I explained to you in class, these two species differ in that one is diploid and one is tetraploid. And so the difference in ploidy prevents them from interbreeding. But if you look at these animals, if you just pick them up in the field, it's impossible for you to identify which species it is without hearing them call or without looking, for example, at the size of their blood cells, because amphibians have blood cells that still have the nucleus in them. The difference in genomic content makes their blood cells slightly different sizes, and it's detectable under a regular microscope. So in this case, you might invoke a species concept called the evolutionary species concept, proposed by Wiley in 1978, which is a lineage of ancestor-descendant populations which maintains its identity from other such lineages and which has its own evolutionary tendencies and historical fate. And that would be the case with both of these two organisms because they are separated from one another in terms of gene flow, and as a result, you might see them maintaining their own identity and having their own evolutionary tendencies and historical fate. But once again, this becomes problematic when we look at, and this shows you that versicolor and chrysosceles, Hyla versicolor and Hyla chrysosceles, are their own independent lineages when we look at genetic divergence. But the evolutionary species concept becomes problematic when we then look at things like Aspidicilus uniparens. Aspidicilus uniparens is a daughter species that comes from a hybridization event between two parental species, Aspidicilus gularis and Aspidicilus inornata. Now, when these two parent species created this hybrid became an all-female species that reproduces through parthenogenesis. It reproduces by producing new offspring from unfertilized eggs. Because there is no gene flow between members of the species Aspidicilus uniparens, then in a way, each one of these lizards, each individual lizard, is its own evolutionary lineage. And it would be ridiculous to assign an individual species name to every Aspidicilus uniparens out there running around on the landscape, even though there is no gene exchange between members of Aspidicilus uniparens, except through the process of reproduction. And once again, this is parthenogenic reproduction. So evolutionary species concept is not a panacea at all. Then there's the recognition species concept, and this is kind of useful in some ways. A species is a group of organisms that recognize one another as potential mates or that share a common fertilization system. And in this case, those gray tree frogs could be recognized through the recognition species concept, because when a Hyla chrysosceles calls, a female Hyla versicolors knows that that is not their species call, so they can recognize themselves as different from one another and not mate. Another example of this is a situation in spiders. This is Schizocosa ocreata and Schizocosa craspes. These two species are virtually identical in the males and also identical in the females, and the only way in which they differ from one another is in a series of courtship and mating rituals that they go through, the courtship dances that the males perform in order to encourage and induce a female to mate with them. And so in this case, once again, when you're a male spider, you come up to a female spider, you perform a dance. She perceives that dance as either an invitation to mate, or she perceives that dance as, oh, there's some food moving over there. And in the case of perceiving it as courtship, she will go and engage in having sex with that male. But if the male is presenting the wrong mating dance, the wrong courtship signal, she will just perceive that as a spider of a different species that is actually a potential food source and will come over and attack and kill and eat the male. And so once again, this is recognition of mating signals that determines the inclusion of individuals in the same species. So when we talk about the Darwin's finches, the Galapagos finches, it's a species complex that is the result of an adaptive radiation. And one of the primary ways in which you tell these birds apart is by their beak morphology. And the beak morphology is important because they're cracking these seeds open to get the ovules inside. And the ability to gain access to these seeds is crucial to their survival. And so what has happened over time is that different species of finches have evolved different beak morphologies that allow them access to different parts of the community of seeds that are out there on the landscape in the Galapagos Islands. So historically, there's this example of rapid change in Darwin's finches back in the 1970s. And so this is, once again, Geospiza fortis. This is the pre-selection distribution, a mean of a little bit less than 9.5 millimeters in terms of beak depth. This is a ton of birds, 751 birds go into this histogram. And then what happened was this was at the peak of an El Nino when there was a lot of rain, there were a lot of seeds, there were a lot of birds. After the El Nino went away, there was then a drought. As that drought proceeded, the population dropped drastically in terms of the number of finches, and the morphology of the seeds changed from being kind of an intermediate-sized seed to becoming a very hard seed to becoming a very soft seed in terms of overall. Oh, I'm sorry, this is abundance of seeds. Went from being a lot of seeds to very few seeds after the drought was over. And what happened during this is the hardness of the seeds changed. Went from kind of average seed hardness to a dip in seed hardness to an increase in seed hardness, and then kind of back down to small and soft. But during this time that the population was decreasing, the population of seeds was decreasing, what that meant was that only the hard seeds were left behind. And as a result of this, because you need a big beak in order to crack these large, hard seeds, what you saw was a shift from before the drought in 1976 to after the drought in 1978. There was this evolutionary shift in beak size from a little less than 9.5 to a little bit more than 10. Also look at the numbers. These are all the Daphne birds, and these are the ones that survived, and there are many fewer of them in the post-selection population. If we look at heritability of of beak depth, we have offspring beak depth on the y-axis, mid-parent beak depth on the x-axis. Once again, this correlation, the slope of the correlation is the measure of heritability. In both 1976 and 1978, the trait is heritable, but you'll see that the average size of beaks in offspring and parents is lower in the 1976 blue regression, and both adult mid-parent beak and offspring beak depth are higher in 1978. The other thing that is interesting about this is that you see that in the 1978 data, there are no beaks that are less than about 8.8 millimeters in length, and so you've lost this low end of the population. You will also see that this slope here is also slightly less steep than the slope before, and once again, that slope value, because it is lower, it means that you're now less heritable because you've lost some genetic variation, because you've lost that low end of the trait distribution, and this is exactly what directional selection does. And so what has happened over time during this time period is you went from regular beaked finches up to very large beaked finches, and then over time that slowly went back to being medium beaked finches as a result of just slow drifting of this population going back, probably in the lack of directional selection keeping those beak sizes large. So this is an example of microevolution, evolutionary change within a species that doesn't result in the production of new species. This is a morphological change within a particular species, Geospizophorus. We're not talking about any other species except Geospizophorus here, and it fluctuates in average beak size within this biological population over time. So this is kind of what it looks like. Over time, the phenotype may change, but there's no branching, there's no production of new species from this. And it may be that after a period of time, that species looks somewhat different from the species that was at the beginning, but once again, there's been no production of new species here. Macroevolution, on the other hand, is evolutionary change that does result in the production of new descendant species. And so what happens in this case is the phenotype changes, but there comes some point where the population divides into two subpopulations, and when those subpopulations evolve away from one another, they finally become different enough morphologically that individual biologists will start recognizing these two evolutionary lineages as individual species. And this is exactly what we think has happened with Geospizophorus, Fulginosa, and Magnorostris. We have bill depth once again here, where Fulginosa is a small beaked finch, Fortis is a medium beaked finch, and Magnorostris is a large beaked finch. This is where they all occur together on the same island, but there are some islands where only Fortis occurs, and there are other islands where only Fulginosa occurs, Santa Cruz and Daphne Major. And on these islands, you see that both populations are closer to the midpoint between them than they are on islands where I'm sorry, I have this wrong. On Santa Cruz, they're all three there. On Daphne Major, it's only Fortis that is there, and on Los Hermanos, there's only Fulginosa. So on these two islands, Daphne Major and Los Hermanos, where they occur by themselves, their phenotypes are much closer to one another than on the island where they occur together. And this character displacement, this movement of the phenotypes away from one another, is due to presumably what Connell refers to as the ghost of competition past. You see this divergence in morphology because that is presumably the fingerprint of previous selection that has been brought on by competition, in this case competition for food. So we have other examples of speciation going on. A really good textbook example of speciation going on is the ring species of Insetina escholzi, which is this salamander, a plethodonid salamander, once again, that lives along the coast range and then into the Sierra Nevadas. Presumably at one point there was a continuous distribution of these, but as the Central Valley dried up, it separated these populations into geographically isolated populations, and these animals have kind of spread down the Sierra Range and down the Coast Range, so that there's a continuous interchange of genes from this species to Platensis, Platensis to Oregonensis, Oregonensis to Picta, Oregonensis to Xanthoptica and Escholzi. But these two species down here at the bottom, the one illustrated in pink, which I'm not sure how they pronounce that, croc-eater or croquiator or something like that, and Escholzi, are incapable of interbreeding because they're separated by all of this evolutionary time that it took for the two arms of this species to grow down and meet here in the middle. And so this is a ring species, a continuous series of populations that are capable of interbreeding with one another, but the ones at the endpoints are incapable of breeding with one another. So modes of speciation, how do species go about forming? We can have sympatric speciation, which is speciation that occurs without the imposition of geographic barriers to gene flow. A good example of this is this paper by Rice and Saltz from 1989. They did this in a lab. What they basically did was they gave fruit flies a choice. They would put the fruit flies on these tubes in the middle. They would go into this tube, and then they had the choice. They could either fly up or they could fly down. And they could go to the light side or the dark side. So actually, this is really the choice. They're put into the central tube. They can go to the dark or they can go to the light. Once they're in the dark, they can either fly up or they can fly down. Once they're in the light, they can fly up or fly down. Once they've made that decision to fly up and down, they can choose to go down a tube that is scented with ethanol or a tube that is scented with acetaldehyde. And as a result, they kind of self-segregate. Population has individuals. Some individuals say, oh, I like the dark more than the light. They go into the dark. Some individuals say, oh, I like to go up rather than to fly up rather than fly down. So they fly up. Once they've flown up, they then choose ethanol, which is the dark tube, or acetaldehyde, which is the light tube. When they did this, they allowed these animals to breed and lay eggs. Then they took these tubes off, let the adults go away, and then put the fertilized eggs back into this apparatus. When those individuals hatched out, they went into this tube. They chose the light or the dark. They chose the up or the down, and they chose the ethanol versus the acetaldehyde. They did this generation after generation after generation, and they had a morphological marker to track individuals, and that morphological marker was the presence of brown eyes. And they basically looked to see what the proportion of brown-eyed flies was in this vial and this vial after so many generations of allowing flies to kind of self-select. And this is go to the dark, go up, go to acetaldehyde. This is go to the light, go down, and go to ethanol, essentially. And when they plotted these, the tops are the experimentals where there were actually differences in all of these things, and the bottom are controls where these differences, the same apparatus was there, but there was no difference between any of these things. And you can see that these populations are diverging over time, and this was once again done over the course of 25 generations. So they're all sympatric. They all come together back here in the center, and then they basically just choose different habitats within this larger habitat, different microhabitats within this larger habitat to breed. And once they get there, they breed with other individuals who have made their same choices, and so those choices become reinforced over time. This is very different from allopatric speciation after the imposition of a barrier to gene flow. And so gene flow barriers can be set up because organisms disperse across some barrier, and the success of that dispersal across the barrier is not very common, and as a result, there's just generally low levels of gene flow between the parental population and the founding population. The other thing is that barriers can encroach upon the distribution of widely distributed species through the imposition of the change in the course of a river, or mountains rising, or deserts forming, or any of these things can produce barriers to dispersal. It isn't that the population moved, it's that a barrier to dispersal arose that then separated the population into two or more segments. So those are the two kind of models of speciation, allopatric speciation and sympatric speciation. But however it goes about it, speciation can result in adaptive radiations. So the rapid diversification of multiple species from a common ancestral species, often occurring after a species first colonizes a new environment, is what adaptive radiation is. So I want you to pause the video here for a minute and think to yourself, from a resource competition perspective, and Dr. Judd has been talking to you about resource competition, how would colonizing a new environment facilitate adaptive radiation? What do you know about competition, the availability of resources, and what happens to organisms under resource competition that makes colonizing new environments a situation that would allow adaptive radiation to occur? And you need to think about this both in terms of competition, but also think of this in terms of density-dependent and density-independent population growth models. So take a minute, think through that. Now you've paused, you've thought through that. So in a colonization model, what you see is a new individual or a couple of individuals colonize a new island. When you arrive on the island, you have all the resources of that island to yourself. And so you can ask yourself, well, what would a species do under those conditions from the standpoint of population growth? Well, you should predict that this population would undergo exponential population growth. It would basically fill the ecological space on the island. But once you get so many individuals on that island, you then begin to have density-dependent factors starting to kick in, such that some individuals may survive better than others. Well, if that survival depends on their morphology, you have a situation set up to have selection acting. As this exponential growth occurs, these individuals expand to new habitats. Because there's now competition, when they get into these new habitats, they adapt to local environmental conditions, and this causes them to undergo disruptive selection. One subset of the population in one area does things one way, another subset of the population in another area does things another way. And over time, these subpopulations will slowly diverge from one another. So there are lots of examples of adaptive radiations. The anoles in the Caribbean is one good example of this. And in this case, there appears to be a series of island-specific adaptive radiations that have gone on. This is from Jonathan Lose's book, Lizards in an Evolutionary Tree, that basically shows the different kinds of anoles that occur on any given island. You have crown giants, you have trunk crown anoles, you have twig anoles that come out on these bare twigs at the edges of the canopy. You have trunk anoles, you have trunk ground anoles, and you have grass anoles. These are all what we refer to as ecomorphs. They have a morphology that is suited to the ecological place that they occur in the canopy of a tropical forest in this case. In Puerto Rico, we have, for example, a trunk anole, the Anole Ecomorphs. We have a twig anole that occurs most commonly up in the canopy. We have a highlight gap specialist. It tends to occur in trefoil gaps. This is Anole Evermanni. And we have a grass anole, which occurs, once again, in light gaps and in grassy environments. This is Anole krugi. There's another similar species, Anole pulchellus, that looks very similar to this guy, also on Puerto Rico. And then, of course, we have the crown giant. Of all the time that I spent in Puerto Rico, I've only seen one crown giant. This is Anole cuvieri, and it's easily 3 times bigger than the next biggest lizard, which is Anole ecomorphs here. And so you can see how big its head is relative to the thumb of this was a graduate student who was doing some survey work nearby, who also took this picture of me with the only Anole cuvieri I've ever seen. So what has happened, if you look at this paper that was published in 2017, where they've done a phylogenetic analysis of all of the anole species in the world that are still around, you can see that these things are coded by island. So we have the northern Lesser Antilles, we have the southern Lesser Antilles. This is the root of the tree, and the tree goes down, and right here the tree has to break, and this break here is connected to this break up here. So this is just one big tree that has been broken into 2 parts to fit into a figure. What I want you to concentrate on is this section here and this section here, and so I've put those on the next slide. And when we zoom into this, what we see is that Cuba, C stands for Cuba, J stands for Jamaica, NLA is the Northern Lesser Antilles, H is Hispaniola. And we can see that for each of these islands, for example, Puerto Rico, where I've worked in the past, we see that all the Puerto Rican species, for the most part, are members of a single clade. Here is a Cuban clade, here is a Jamaican clade. And you'll see that there are a series of Cuban clades. There's this Cuban clade here, there's this large Cuban clade here that encompasses a lot of different species. There's a Hispaniolan clade, there's another Cuban clade here. And so Cuba, it makes sense that Cuba might have multiple adaptive radiations because it's a fairly big island relative to Hispaniola and Puerto Rico in particular. And Jamaica is also very small. And so you might imagine that different species arrive in Cuba at different times, maybe arrive in different parts of Cuba that are not occupied by lizards, and so you have the opportunity for multiple adaptive radiations going on within Cuba, whereas you have single adaptive radiations on Hispaniola, Puerto Rico, and Jamaica. The other thing that this kind of illustrates to you is going back to island biogeography. In island biogeographic models, one of the things that you know from that is that island size is an important factor in determining how many species an island can possess at the equilibrium level. And you see that Cuba has a lot of species from these 3 different radiations. Hispaniola has fewer species than Cuba, but more species than Puerto Rico. Puerto Rico has more species than Jamaica, but fewer species than Hispaniola, and that's the order of size. So this is also phylogeny that also speaks to the theory of island biogeography. And then lastly, we have the Darwin's finches. So here is the adaptive radiation of Darwin's finches. This is a 2013 figure. Since then, for example, they have divided up the sharp-billed finch into 3 different species, one of which is a species that's sometimes called the vampire finch. It pokes on lizards and drinks their blood, essentially. But once again, a big way in which these groups can be separated from one another is in bill morphology. And so we have at least 14 species, maybe 17 or more species that have all been divided up, and it shows you the type of beak that they have, and it shows you the type of diet that they have. And you can see that there's a close correspondence to the type of beak and the type of diet. And so Hendry's ideas that the food that is available on the island is very important in determining which types of beak morphology survive and reproduce. That's the thing that is the basis of selection among these different bird species. And so you're looking at the medium ground finch that is basically eating these types of seeds, and as a result of that, any variation in the morphology either equips you to eat bigger seeds if you're more towards Magarostra size, or smaller seeds if you're more towards Fuliginosus size. And you can see that these three species are all more closely related to one another than they are to anybody else in the tree, which then goes back to this figure that we had some time ago looking at character displacement. These three species that all are closely related to one another also constitute species that, when in the absence of the other species, they tend to move out of the morphological space that they're in when they're found together on an island. Fortis has smaller beaks on islands where they're by themselves. Fuliginosa has larger beaks when they're on islands by themselves relative to when they are together with another species. And with that, hopefully that takes you through a little introduction to speciation and adaptive radiation. Thanks for listening.